Hi everyone, my name is Sakshi Mohta and I'm really excited to share my research on the topic computational drug repurposing to treat schizophrenia using network medicine. So I look forward to discussing these findings with you and exploring how we can further advance our understanding in this area. Introduction. So the process of developing new drugs in general is extremely laborious, expensive and time consuming. It is even more difficult to treat PNDs as they share a large number of clinical and genetic characteristics, which makes it more challenging. A serious mental illness that alters the perception of reality, schizophrenia. It affects 23 million people worldwide. Drug repurposing, however, is a recent alternative technique. The repurposed drug has previously been approved by the FDA, so the economic burden of testing for tolerance, safety, side effects, efficacy and regulatory affairs associated with phase 1 trials is reduced, making it a cost-effective approach. The recently developed field of computational network medicine can prove to be very promising as it applies the concepts and tools from network theory in order to develop meaningful computational approaches for drug repurposing. Materials and methods. Data retrieval. Genetic data was curated from databases such as SZDB and NCBI. PPI data was curated from string. Drug target interaction data from Drug Bank and DGIDB. Network construction and analysis. The list of schizophrenia-associated genes and their interactive data imported into Cytoscape to generate the network. Degree and closeness centrality measures were analyzed. Functional gene enrichment performed. Hub genes were selected as input seeds to generate new protein-protein interactions and new functional associations using string. Network of drugs interacting with only the target hub proteins and their new functional partners was followed by network analysis. Result. The first dataset contained 119 hub genes and functional gene enrichment was performed on the selected nodes using KEGG. The second dataset, it contained 38 hub genes and functional gene enrichment was performed using enrichR in order to identify the biological processes associated with the input genes. In the figure you can see a network of 119 hub genes which are connected via 434 edges. The node color represents the expression of the genes in the nervous system. Yellow colored nodes represent the lowest expression of the genes in the nervous system, and the pink colored genes or the pink colored nodes, they show the highest expression in the nervous system. The light blue color of the border, it represents adrenergic signaling. Dark blue, dopaminergic synapse. Light green, it represents growth hormone synthesis and secretion, and dark green, it represents sphingolipid signaling pathway. Pink represents cholinergic synapse. Protein-protein interaction network in the first figure. The string network shows the second shallow functional partners linked to the input, which includes 119 seed genes. In the second figure is a bar graph which represents functional gene enrichment of 38 identified genes, schizophrenia genes, from the second dataset. In the figure you can see a protein-protein interaction network of 77 nodes connected through 160 edges. Finalizing drug repurposing candidates. Candidates with a high topological measure were primarily used for indications other than schizophrenia. Those were considered relevant for further prioritization. You can see the snapshot of amoxapine which is interacting in the drug target interactome and a snapshot of desipramine which is interacting in the drug target interactome. Prioritized drug repurposing candidates for schizophrenia can be shown in the first table. Desipramine, amoxapine, dasatinib, trazodone and neratinib. Connectivity map analysis. So, a standardized metric with a range of -100 to 100 is a CMAP connectivity score. A query yields a rank-ordered list of CMAP signatures sorted by their connectivity scores using CMAP signature similarity. The following input queries were used to find similar perturbations of chemical compounds. haloperidol, chlorpromazine, flufenazine, etc. 2 out of 5 shortlisted drugs that scored more than 90 appeared in more than 4 query search results. Amoxapine was found among 6 query searches as a top-ranked similar perturbagen to these AZ-approved FDA drugs. Clinical and literature trial evidence was also used to validate my result. I found out that neratinib was identified as a potential repurposing candidate in some literature-based studies. So in conclusion, I can say that despite the challenges such as data scarcity and lack of validation models, techniques like network medicine provide robust frameworks for drug repurposing. Continued innovation in network-based methodologies is expected to advance CNS drug discovery. Integrating diverse data sources and refining algorithms will improve the identification and validation of promising candidates, giving new hope for better treatments and outcomes.